Hello, my friends, and welcome to our video lecture on topic A2.3, viruses. Our guiding questions are, how can viruses exist with so few genes, and in what ways do viruses vary? And our objectives, we're going to describe some features that are common to viruses, and we're also going to look at the enormous diversity amongst those viruses. We're going to look in some detail at a couple examples of viruses, including the bacteriophage lambda, coronavirus, and HIV. We're going to describe a couple of the life cycles of viruses, and I have this in quotes because viruses aren't living, so it's hard to say that they have actual life cycles. We need a better name for that. We're going to look at some of the theories of origins of viruses and talk about the convergent evolution amongst viruses. We're going to talk about how fast that evolution occurs in many viruses, and then we're going to take a look at antigenic drift versus shift in that evolution. Before we dive into details related to the structure of viruses, we're going to talk about some of the vocabulary related to consequences of viral infections. Outbreaks are when we have an increase in the rate of infection. Epidemics are when those outbreaks spread to large geographical areas. Pandemics, when those infections spread globally. We've had quite a few pandemics over the course of human history. Of course, the bubonic or Black Death Plague in the 1300s, well-known pandemic, the Spanish flu in the early 1900s, HIV and AIDS pandemic started happening in the 1980s and continues through today. And then, of course, most recently, the COVID-19 pandemic. Quite a few of those pandemics were caused by viral infections, amongst them HIV, the Spanish flu, and COVID. Viruses are incredibly diverse. There are only a few commonalities amongst all viruses. One of those commonalities is that viruses are all very, very small. They range in size from 10 to 400 nanometers. To compare, a typical bacteria cell is about 2,000 nanometers long. So these guys are super small. They have fixed sizes. They don't grow. They are simply assembled. They're going to have some kind of genetic material. That genetic material might be DNA, it might be RNA, it might be double-stranded, it might be single-stranded, it could be linear, it could be circular. So a lot of variation within that genetic material, but all viruses do have some kind of genetic material. All viruses also have a protein coat that we call a capsid. Those capsids are the boundaries between the virus and the environment. Those capsids protect the genetic material on the inside. Sometimes those capsids will be surrounded by a membranous envelope formed from the cell membranes of their host cells. Sometimes there's no envelope, but all of these capsids allow for the viruses to bind to host cells, very specific host cells, so that the genetic material of the virus can be inserted into those host cells. Viruses have no cytoplasm. They have very few, if any, enzymes, because what happens is the genetic material of the virus is inserted into the host cell. These guys are parasites, and then they take over the metabolism of the host cell. So the great majority of the work done to reproduce viruses is done by those host cells. Because so much work is done by the host cells, that means that the genetic material of the viruses does not have to be very large. Most viruses have just about 100 genes, which is quite small. E. coli, another bacteria, has 4,000, more than 4,000 genes. So very little genetic material because of that host cell metabolism takeover. As you can see on the slide, there's a great variation in the shapes of viruses. We could have a thread-like or helical shape of a virus. This guy is called a polyhedron. This one is spherical, and this is a bacteriophage. I'm not really sure how to describe that shape. It's, I don't know, like a little spaceship or something. Good. All these different viruses are going to bind to different host cells. Tobacco mosaic virus binds to a protein in plants. Adenoviruses that cause the common cold bind to an immune response protein called CD46. The influenza virus binds to a specific kind of acid domain in lots and lots of membrane proteins, which is why the flu can infect so many of us in so many ways. Bacteriophages are going to bind to a special protein in bacteria cells. Bacteriophages are viruses that infect bacteria. Other examples of viruses: the poliovirus infects nerve cells, hepatitis infects liver cells, norovirus infects intestinal cells. We're going to look at 3 examples of specific viruses. The first one that we're looking at is Bacteriophage lambda. Here is an electron micrograph image of this virus. This is a virus that infects bacteria. It has a capsid protein coat head. Inside that capsid is DNA. This is known as the tail sheath. It has a tail tube that can actually be inserted through the membrane of a bacteria cell. And then these are the tail fibers that help to bind the bacteriophage to the cell membrane of the bacteria cell fibers. We can see here those tail fibers bound to the cell membrane of this bacteria cell. The tail sheath is going to contract, and that allows it to drive the tail tube through the bacteria cell membrane. This particular bacteriophage lambda binds to the lambda B protein of the bacteria cell. And then the DNA that is stored here in the capsid can just get pushed right on inside the cell, and then the virus DNA takes over the metabolism of that bacteria cell. And our second example of a virus is the coronavirus. COVID-19 is our most recent pandemic, at least as of 2024. This stands for coronavirus disease of 2019. SARS stands for severe acute respiratory syndrome. Coronavirus number 2 is the specific virus that led to COVID-19. We call these guys coronaviruses because they have these amazing spike proteins embedded in the envelope of the virus, and in electron micrograph images, it makes it look like it has a crown, hence coronavirus. Coronaviruses have some single-stranded RNA inside them. They have, of course, their protein capsid coat that helps to protect that RNA. They also have an envelope composed of membrane that it stole from some host cells, and embedded in that envelope are those spike proteins, the spike glycoproteins. These guys bind to a specific receptor commonly found in us humans in our respiratory system. That receptor is called ACE2, or angiotensin converting enzyme number 2. The third and final example of a virus that we are going to look at together today is the human immunodeficiency virus, also known as HIV. HIV, like coronavirus, does have an envelope, that lipid membrane envelope. It's got its capsid on the inside. It also has RNA, just like coronavirus. What's a little bit different is that HIV actually has 2 strands of RNA, both single-stranded. These guys are identical to each other, so those 2 RNA strands exactly the same. Coronavirus only had that 1 strand of RNA. There is some enzyme in human immunodeficiency virus and HIV called reverse transcriptase. One of the genes in the RNA actually leads to the production of more reverse transcriptase. What's kind of interesting about HIV is what we call a retrovirus. That RNA is actually reverse transcribed back into DNA. That DNA is inserted into the human genome, and then that DNA is used to make messenger RNA, which is then translated into viral proteins. This process, reverse transcription, is full of mutations. It's not super high quality, which leads to a lot of mutations in these retroviruses. We'll talk about that a little bit more later. This guy also has these super cool glycoproteins on its envelope. This is going to allow HIV to bind to specific receptors on human T cell lymphocytes. These guys are cells involved in the immune response. We're going to look next at two cycles that viruses go through, and we call them life cycles, but again, viruses are not living, so this is kind of a bad name for this. The first cycle that we're going to look at is the lytic cycle. The first step is absorption or attachment, where the virus attaches to the host cell. We then have penetration and the injection of the DNA or the RNA into the host cell. We're going to go through the biosynthesis of new pieces of the virus, and then we're going to have the maturation of the virus when those pieces and parts get put together to assemble mature viruses. The host cell is then going to lyse. That means that it breaks apart, it splits, explodes open, and all these new viruses, we're going to call those virions, are released. Again, these guys can go and then attach and penetrate, biosynthesis of new pieces of the virus, that maturation or assembly of the viruses, and then lysis again. The second cycle that we're going to look at is the lysogenic cycle. In lysogenic cycle, we still have that virus attaching. It is going to penetrate and insert its DNA. But what's going to happen is not right away a production of new viral particles. Instead, the piece of the viral DNA is called a prophage. That prophage is inserted into the host cell's genome. And here we're looking at binary fission of the bacteria, but if this were a human cell that was being infected, then we would have some mitosis happening. And what's interesting is that each of the daughter cells produced in mitosis or binary fission has a copy of the viral DNA. So now we have a whole population of cells that include that viral DNA. At some point, some trigger is going to cause the excision or release of the viral DNA from the chromosome, and then it becomes active. Then we're going to start to make lots and lots of pieces of virus, that biosynthesis, the maturation of the viruses when we assemble those new guys, and then we'll also have lysis. Except it's not going to be just one cell. It's going to be all of those daughter cells that were produced during binary fission or mitosis that all have copies of the viral prophage in them. This means that there's going to be a huge impact, often quite suddenly. We don't actually know exactly what those triggers are at some point. We don't really know what those some points are. We think that perhaps UV radiation has some effect on this excision of the viral DNA. We think that there might be some chemicals that lead to the excision of this viral DNA, but we don't really know completely yet. So if you are interested in doing research into disease, this could be a super cool place to investigate. So where did all of these viruses even come from? We don't actually know that either, but we've got quite a few theories. The first one is the virus first model, which states that because viruses are more simple than our biological cells, perhaps viruses actually evolved first. And perhaps those ancestors of our modern viruses actually provided some of the pieces and parts needed to have those biological cells evolve. We also have an escape hypothesis. Perhaps we had a cell that had lots of stuff, including some genetic material, and some of that genetic material escaped from the cell in a little vesicle, went through some self-replication, and we ended up with viruses. There is also the degenerative model, also known as the regressive model. And this states that we had a couple different kinds of cells, but one of them, this little guy, kind of lost a lot of its pieces and parts and then became a parasite on a larger cell since it had lost so many of its pieces and parts. What we do know is that the human genome has at least 8% from viruses, so 8% of our DNA is actually viral DNA. We also know that viruses likely played a huge role in evolution of this species that we have on Earth today. We also think that perhaps viruses evolved multiple times. We might have had different origins of different viruses at different times, but over the course of Earth's history, some convergent evolution happened. Convergent evolution is when we have things that start looking different, but then over time mutate and evolve to look more alike. So in these pieces of letters, I'm just kind of showing that perhaps we had two different things, A's and T's, and over time they evolved to look like each other. Those S's look pretty similar. So it is possible that we had a lot of different origins of viruses, and over time they've just come to look more alike. How they look alike, they are all obligate parasites. They must have a host cell. They cannot reproduce on their own. They all have some kind of protein boundary. They're capsid. None of them have cytoplasm. They all have that genetic material, RNA or DNA. It's hard for us to know a lot of things about the history, the evolution of viruses, because they don't make fossils. So it's really hard for us to look at them in the past and just know things. One thing we do know very well is that viruses evolve incredibly rapidly. The reasons for this, they have very large population sizes. They have very short generation times. Viruses don't last long. They are reproduced very quickly. They also have a crazy high mutation rate. This graph is showing some of the mutation rates of some different viruses. Those viruses are listed here. The mutation rate is how many mutations per base per cell. So here we're looking at 1 out of 100, 10 to the minus 2 is 100. Here we're looking at 1 out of a million. And notice that HIV in vivo, this is in living things, has a crazy high mutation rate. HIV that is in cell culture, so not in living things, has a slightly lower mutation rate, but both really, really high. So these viruses are incredibly quick to mutate. This makes them really hard for us to have vaccines for. One of the reasons that HIV has such a crazy high mutation rate is because it is what we talked about a little bit earlier, a retrovirus. So this retrovirus, HIV, is going to insert its RNA. That RNA is going to be reverse transcribed into DNA. This process is not very highly controlled. And so many errors are made when we copy the RNA to DNA. This leads to lots of mutations, and then these viruses are going to look a lot different from those original viruses just because that mutation rate is so very high. And we have two ways to describe this evolution of viruses. We can talk about antigenic drift and antigenic shift. An antigen is the part of a pathogen that our immune system recognizes. Pathogen is a disease-causing agent. The antigen is a particular piece of the pathogen that our immune system goes, you don't belong here, let me fight you off. Antigenic drift is the drifting of these antigens, how they can change a little bit over time. Very small changes over long periods of time. That's going to lead to a slow variation in the surface proteins. But if those surface proteins are changing after a while, the accumulation of those changes might prevent our immune systems from recognizing those viruses, and then we don't know how to kill them off anymore. An interesting thing about HIV, HIV goes through antigenic drift. There are small changes over long periods of time, except those actually happen really, really fast. So we have small changes, we have a slow variation, but we don't have a very slow variation because there are so many changes, even though they're small changes, that HIV actually drifts quite rapidly. Sometimes we can have our vaccines be adjusted and still be effective as antigenic drift happens. That doesn't work for HIV because it just drifts too fast. The other kind of evolution that we look at is antigenic shift. This is where, are you ready for this? We have two or more viruses that infect the same cell, and then they mash up together. They combine their genetic material, which leads to a huge change in a very short amount of time. That rapid major change in the surface proteins leads to a totally new viral strain that our immune system just doesn't recognize anymore. Influenza, the flu virus, is really good at this. And we can try to adjust our vaccines, but they usually aren't very effective since the changes are so large and so fast, which is why we have to get a flu vaccine just about every year as opposed to chickenpox, which, one and a booster and you're done for generally life. And on that note, my friends, we are done with this video lecture on viruses. We talked about those features that are common to viruses. They are small, they've got fixed shapes, they've got some genetic material, but no cytoplasm and no organelles, no enzymes, maybe just a few enzymes. Lots of diversity in terms of shapes, in terms of how they bind to their host cells, whether they have DNA or RNA. We talked about bacteriophage lambda that infects bacteria, coronavirus, and HIV that infect us humans. Those life but not life cycles of viruses, because viruses are not living, can be lytic or lysogenic. We talked about theories of origins of viruses, that escape theory, the regressive theory, the virus first theory. We talked about convergent evolution of viruses, how over time they've started to look a lot alike, those common features. We talked about how evolution of viruses can be very rapid because they mutate so fast. We talked about antigenic drift versus shift. Great work today, my friends.